Hello everyone. Welcome to today's live broadcast, The Benefits and Burdens of Assaying Matched Normal Tissue when Sequencing Cancer Genomes. My name is Toby Ward and I'm a Field Application Scientist at Personalis Incorporated, and I'll be your moderator for today's event. We're pleased to bring you this educational web seminar presented by LabRoots and sponsored by Personalis. We are a full solution genomics sequencing company in Menlo Park, California. For more information, we invite you to please visit personalis.com. We do have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. I will queue these questions as they come in, and we'll try to answer as many questions as we can by the end of the presentation. If you do have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. This is an educational webinar and thus offers free continuing education credits such as PACE, RACE, CME, or CEU. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page, and you can follow the process to obtain your credits. With that, I would now like to introduce today's speaker, Dr. Elena Helman. Elena joined Personalis over 2 years ago as the technical lead in our bioinformatics science department and has been instrumental in leading our development of cancer sequencing and analysis pipelines. Throughout her career at the Broad Institute and at Harvard and Brown universities, Dr. Helman has led cutting-edge projects in cancer bioinformatics, including designing tools to detect genomic rearrangements and structural variants in whole genome and whole exome cancer sequencing data. Today, Elena will discuss the benefits and burdens faced when sequence data from a matched normal tissue is analyzed in parallel with a tumor specimen. The learning objectives for the presentation are, first, to understand the impact of a matched normal sample on the analysis pipeline, the data filtering, and the database usage for the project. Second, to consider the burden of secondary findings when sequencing a matched normal tissue from an individual. And finally, to appreciate the definition of actionable variance in your study design and how this impacts the results from the analysis. With that, I'd now like to turn it over to Elena for her presentation.
Thank you, Toby, for the introduction, and thank you to everyone for tuning in with us this afternoon as we discuss our investigation of some of the benefits and the burdens of sequencing a matched normal sample when assaying a cancer genome. Now, I'll start first with an overview of Personalis, who we are, what we do, and describe our proprietary ACE sequencing technology. I'll then introduce the different products we've built to solve some of the challenges involved in cancer research, in clinical trials, and in immuno-oncology applications. Then I'll discuss our validation strategy for small variants using our ACE cancer exome. And finally, I'll dive into the bulk of our presentation, which is to discuss, again, the benefits and burdens of using a matched normal. And lastly, I'll be happy to take any questions at the end. So Personalis was founded in late 2011 by our CEO, John West, and our scientific founders, Stanford professors and physicians Russ Altman, Mike Snyder, Atul Butte, and Ewan Ashley. We hold multiple patents, both in the US and in the UK, for our proprietary ACE technology. And in 2013, we launched our first clinical test, the ACE clinical exome, which was for inherited and Mendelian disorders. But since then, we've really had a strong focus on developing our cancer portfolio. We have both a CLIA and a CAP accredited laboratory, high-performance computing clusters, and a strong bioinformatics and operational expertise in-house. Now, Personalis was founded really to address the major challenges in next-generation sequencing. Our mission is to develop novel methods to increase the accuracy at each step in NGS and interpretation. So some of the issues associated with sample prep and sequencing include handling of varying sample amounts and qualities, as well as suboptimal coverage. We've also spent considerable effort in optimizing our analytical pipelines to address some of the inconsistencies we found that are associated with the reference sequence, with imperfect alignment, and of course with somatic variant calling. We've also collected a large compendium of databases and annotation resources in-house, including over 40 public and proprietary sources. And we've really taken the effort to standardize and normalize all of the data coming in from all of these discrepant sources, which really is no small task. So at each step of this process, we've focused on creating novel and innovative solutions to increase the accuracy and robustness of our end-to-end NGS solution. Now, one of the biggest challenges in sequencing today are the gaps and the inconsistencies with standard assays like a standard exome. So for example, we show here a coverage plot of the STK11 gene using a standard exome. The plot shows all exons concatenated on the x-axis and the read depth on the y. And so as you can see, although this gene is sequenced at high depth, in this case at an average of over 100x, the standard exome fails to cover entire exons. So variants residing in these regions would be completely missed. So to address these gaps in the standard exome, we at Personalis have developed our ACE technology, and ACE stands for Accuracy and Content Enhanced. We've implemented a series of both probe design steps as well as optimization of sample library prep to augment coverage. And we've built an ACE exome design that works by taking the standard exome, here in blue, and adding supplemental coverage in green. And these are combined in an effort to maximize the genomic content and provide more uniformity in coverage, which increases confidence in calling variants in these regions. Not only do we target exonic content, but we've also augmented coverage of biomedically important regions outside of the exon, such as regulatory regions, intergenic and intronic areas, as well as difficult-to-sequence regions of the genome, such as GC-rich areas. So when we look again at our STK11 example, we have now our augmented ACE coverage, shown in green, in addition to the standard exome coverage, which is again in blue, and now this provides 99% coverage of exonic regions in a very uniform manner. So with ACE, clinically important variants residing in these hard-to-sequence regions can now be identified. Now we apply this augmentation to 8,000 medically relevant genes, including 1,400 cancer-relevant genes across the exome. So for an application such as mutational load assessment, we can use our enhanced exome for both broad and highly accurate profiling of the mutational landscape of the tumor. So these sequencing plots shown here show ACE performance, again in green, for PRDX5 and ACTN4 genes. Both of these genes have been shown to carry somatic mutations that may contribute to the mutational load in these types of cancers. And again, variants residing in green regions would be missed with the standard exome, but with our ACE exome filling in these gaps, we can now get a true representation of the genomic landscape for each cancer sample. We apply our ACE technology to a number of different cancer offerings. We have an ACE extended cancer panel for both DNA and RNA. We also have a clinical version of this test for tumor characterization and clinical interpretation. Then we have our augmented ACE exome and transcriptome. And all of these assays are designed to aid in cancer research, in clinical trial investigations, as well as in immuno-oncology research. Now for this webinar, I'll be focusing on two of our assays more specifically, the ACE cancer gene panel for DNA and our ACE cancer exome. So I'd like to delineate the differences between the two here. Our ACE cancer gene panel targets over 1,400 cancer genes. Now, we designed this panel to try to assay almost all of the genes a cancer investigator might be interested in. This includes, of course, all actionable cancer genes and hotspot mutations, as well as genes commonly mutated across cancers, as described by the Cancer Gene Census and other large studies such as the Cancer Genome Atlas. We also target genes that play a role in canonical pathways of cancer, as well as over 200 cancer-related microRNA genes. Now, this large target region is then sequenced to an average of about 500x coverage for high-depth accuracy. Now, our ACE cancer exome, as I've discussed, is a full standard exome with the augmented coverage of cancer content per our ACE technology. The exome is typically sequenced to 150 to 200x depth and is a great resource for large discovery applications. Now, for our DNA analysis, each sample goes through an extensive workflow, as shown here in the diagram. Somatic variants, including SNVs, indels, MNVs, and somatic CNAs, are reported, as well as somatic variant annotations and relevant plots, including genome-wide copy number and mutational signature. And for all of our cancer assays, we think it's important to offer a complete set of deliverables. So we return all raw data, including FASTQ and BAM files, variant calls, and variant annotation. During this talk, I'll be focusing on our DNA analysis, but we do return a full suite of deliverables from extracted RNA as well. And finally, we also return extensive QC reports on the sequencing data with over 100 read, alignment, and variant stats. So it's very easy to determine quickly the quality of the sample of the sequencing at first glance, as well as the overall somatic variant calling statistics. So accuracy is very important to us, as I hope I've made clear by now. In order to assess the accuracy with which we call somatic variants using our assays, we conduct rigorous analytical validation studies to determine our limits of detection. So for the sake of time, today I'll briefly describe our strategy and results from just one of our validation studies, our ACE cancer exome tumor normal workflow. So to determine our analytical sensitivity for SNVs and indel calling, we collected a set of 11 well-characterized tumor cell lines and their matched normal cell lines. We sequenced the DNA from all of these cell lines with our ACE cancer exome and ran the data through our tumor normal bioinformatics pipeline. We then compared our variant calls to a set of gold standard variants to gauge our sensitivity. Now, what do I mean by gold variants? Since gold standard reference samples are not really available for the breadth of the assay that I'm discussing, we curated a gold set of somatic variants using the COSMIC verified events. Now, these events were detected in the COSMIC cell line project in the same cell line as we detected it, and was also verified by two other sources, CCLE, the Cancer Cell Line Encyclopedia, and NCI 60, or was independently confirmed by Sanger. So in total, across these 11 cell lines, there are 875 such gold SNPs and 19 indels. Now, to look at our limits of detection for SNVs and indels, we sequenced 3 pairs of cell lines and their matched normals to establish a baseline. We then created a series of dilutions to represent tumor purities ranging from 5% to 80%, as shown. So for example, taking 5% of the tumor cell line, we mix it with 95% of the normal cell line. This simulates a 5% tumor purity, and so on up to 80% tumor purity. And this allowed us to assess variance at mutant allele frequencies down to 2.5%. So we sequenced all 16 dilutions from this series and ran these through our tumor normal pipeline using the corresponding matched normal. So using these, we calculated our positive percent agreement, PPA, and our positive predictive value, PPV, at certain mutant allele frequency thresholds, as shown above. So we implemented the validation strategy I just laid out and ascertained the following validation numbers for our ACE exome. Overall analytical sensitivity was 99% for SNVs and 95% for indels. For our limits of detection, when we look at all small variants present at a mutant allele frequency at or greater than 10%, we have a PPA of 98% and a PPV of 99%. If we further break these out by variant type, for SNPs we have a 98% PPA and a 99% PPV, and for indels we have a 93% PPA at a 98% PPV, again for variants at MAF greater than or equal to 10%. We also conducted a series of reproducibility and titration experiments. So in total, validation of our ACE cancer exome required sequencing and analysis of over 70 samples and resulted in over 7,000 SNVs and 380 indels assessed. So this just stresses the fact that more well-characterized reference standards are needed for large cancer assays such as ours. So having developed and validated some of our assays, we then wanted to see what's the difference in our pipeline performance depending on whether we have a matched normal sample available or not? Now, as Toby stated, the learning objectives from this portion of the webinar to keep in mind are, first, to really understand the differences in pipelines, in filtering techniques, and use of large population databases necessary, depending on whether or not you have a matched normal available. Second, to discuss the non-trivial considerations of secondary findings, education and consent when sequencing the germline. And finally, we'll talk about how defining your set of actionable variants affects your analysis results. So last year, we saw quite a bit of controversy over tumor-only sequencing, or sequencing just the tumor without using the matched normal for reference. So the current debate over the importance of the matched normal for accurate tumor genetic profiling features 2 main viewpoints. On one side, an important paper from earlier last year concluded that the normal sample is necessary to identify germline variants, and up to 1/3 of actionable mutations they found would have been filtered out if the matched normal had been available. Now, on the other side, other groups have asserted that to identify key cancer-related variants, the normal is not necessary and is logistically often difficult to obtain. So these groups continued to provide exclusively tumor-only sequencing and analysis. So we set out to answer this question: how important is the matched normal? To do this, we collected 87 tumor samples where a matched normal sample was available, either blood, adjacent normal, or both. These samples ranged in tumor type from melanoma to renal cell carcinoma, and in sample type with fresh frozen, FFP, and some cell lines in the mix. We sequence these samples using our two assays I've described, the ACE cancer panel and the ACE cancer exome. And sequencing data from each sample was then analyzed using two bioinformatic pipelines, using our tumor-normal analysis and our tumor-only analysis. Now, for our tumor-normal somatic analysis, we perform sequencing, alignment, and variant calling on the tumor and the normal separately. Then, instead of doing a simple subtraction of normal variants from the tumor, we use aligned reads from the tumor and the normal sample together in our analysis pipeline to more accurately call somatic variants. And with the added information of the matched normal, we're also able to identify possible contamination of the sample and estimate tumor purity more accurately. For our tumor-only analysis, instead of incorporating aligned reads from the matched normal, we employ a series of filters and annotations to the variants called in the tumor to approximate somatic status. These filters include a variety of population frequency metrics, a large panel of unmatched normals, mutational impact, and others. And these filters are unique to the tumor-only pipeline and are just some of the ways we try to informatically make up for the missing matched normal. An important part of our tumor-only pipeline is the population frequency filter. Large studies, such as the Broad Institute's EXACT database, have compiled variants of over 60,000 individuals across multiple populations. Now, we've brought these data in-house, normalized across all of these data sets for accurate annotation, and we use population allele frequency-based thresholds to filter out likely germline variants. Now, in this diagram, I show the difference that these population frequency variance filters make on variant calling in tumor-only mode. So without any filter, we see that over 170,000 variants are called across our set of exome samples. If we implement a 1% population frequency cutoff, we reduce this number by 70%. And an even stricter threshold of 0.04%, determined empirically from the frequency data seen on the right, this decreases the number of tumor-only variants to just 34,000. So the effect of these large databases is quite substantial. Now again, we ran all of our samples through both our tumor-normal and our tumor-only pipelines, and then we compared the variants detected by each. So in this graphic, for each tumor, which is a row on this plot, in blue are the variants called by a tumor-normal analysis, in orange are the variants called by tumor-only, and in gray are the variants detected by both. And as you can see here, there are many more variants that are called with our tumor-only analysis that are not called with our tumor-normal. And these trends apply to samples sequenced both on our broad cancer panel on the right and with our augmented exome on the left. So the few variants that are unique to tumor-normal, in blue here, were likely filtered out of the tumor-only analysis because of those stringent filters that I described previously. But what comprises the much larger proportion of variants that are unique to tumor-only mode? To look at this, we plotted the tumor allele frequency of variants called by both modes in gray, and variants unique to tumor-only in orange again. And it's easy to see that those extra variants called by the tumor-only analysis are likely germline by their allele fraction distribution with peaks at 0.5 and at 1. Now, since in this case we actually had the matched normal available as a reference, we were able to cross-reference the tumor-only variants. So for each sample along the x-axis, the height of the bar represents all variants called in tumor-only mode. And in maroon is the proportion of variants that have evidence in the matched normal as well. So you can see here that there's a large range of tumor-only variants that are actually private germlines. And this ranges from 10% to 80% in some samples. But for most, it's about 60% of the variants that are actually miscalled germline. So even with those stringent filters and population cutoffs that we employ in our tumor-only pipeline, many private germline variants are still being called when the matched normal is not used. Now, although we see a wide range of proportion of germline variants, when we plot the actual number of variants on the right, you can see that the number of variants called in tumor-only mode that are actually germline is very consistent across the samples, despite large variation in total variants called. Now, this has important implications for mutational burden applications, because it shows that somatic mutational load can still be inferred even from tumor-only analysis when the matched normal is not available. So we've been looking thus far at all the variants called across our entire exome and our entire gene panel. But next, we wanted to see how tumor-normal and tumor-only analyses differ when we look at only those variants that are considered actionable. So our first approach to this was to use variants annotated in the clinical knowledge base My Cancer Genome, because they represent a highly curated set of variants with associated clinical information. So we searched for these variants in our results from both analyses. And across 57 samples sequenced on our exome, we discovered 34 My Cancer Genome variants using tumor-normal analysis and 34 such variants using tumor-only. And when we created a Venn diagram of these variants, we see that there was a complete overlap. So the tumor-only analysis did not identify any extra or possibly germline actionable variants in this case. So since this was in contrast with previous findings, we hypothesized that these results may be sensitive to the set of variants considered actionable. So as we all know, there is a spectrum of what can be defined as actionable. My Cancer Genome variants lie on the narrow end of this actionability spectrum. So we repeated this analysis by looking at filtered variants in actionable genes as defined by the Jones et al. Paper, 2015, and as well as by the Target database. So here, using these broader sets of actionable genes, although there is still high concordance between both methods, we do start seeing that the tumor-only analysis calls extra variants that would have been deemed germline if the match normal had been available. Now, in addition to classifying somatic variants, sequencing the match normal allows you to detect germline predisposition variants, such as this VHL variant we discovered, suggesting Van Hippel-Lindau syndrome in one properly consented patient. So if we had not had the match normal in this case, this variant would have been flagged as putatively germline due to its features. Its allele frequency and the type of variant suggests that it's likely a germline. And so this variant would have been recommended for further germline testing. So tumor-only would not have missed this variant per se, but it would have delayed the syndrome determination process. Sequencing the match normal may also reveal secondary findings that need to be handled appropriately. The American College of Medical Genetics recommends that pathogenic findings in a set of 56 genes should be reported. Now, this adds considerable burden on the physician in terms of proper consent and education before and after testing. And classifying germline variants in these 57 genes poses additional burden. So to look at this, we took 30 matched normal samples that were sequenced on the augmented cancer panel, which covers 25 of the 56 ACMG genes, and 20 matched normal samples sequenced on the exome. And we found almost 1,200 germline variants called in these 56 genes. We then had our in-house variant classification scientists go through the filtering and the classification process for all of these variants, and the result was just 4 variants in 3 ACMG genes, including the VHL, as I just discussed, as well as a few BRCA1 and BRCA2 pathogenic variants. And not all matched normal samples are created equal. Adjacent normal tissue may often contain some tumor contamination, which can skew results. So here we took a set of 11 tumors where we had both matched blood and adjacent tissue available, and we ran our tumor normal analysis using each of the normals in turn. And as you can see, there are some variants called in our blood normal analysis, in red, that are then filtered out when using the adjacent normal, because there was evidence for the mutation in the adjacent normal. So here's just an example IGV plot, where the top panel shows a mutation in the tumor. The middle panel represents the blood normal, which does not contain any evidence of this clearly somatic mutation, despite the high read coverage. But the adjacent normal on the bottom panel does have some reads supporting the variant, causing it to be filtered out in that case. So from cases such as this, it's clear that varying filters are necessary to account for possible tumor normal contamination. So in summary, we sequenced 87 tumor samples using both our augmented cancer panel and our exome, and analyzed these with our tumor normal and our tumor-only somatic analysis pipelines. We found that the proportion of variants identified in tumor-only that are actually reclassified as germline when the match normal is available varies widely across samples, but averages around 60%. But the absolute number of variants remains constant across a variety of samples. We saw that when you strictly define a set of actionable variants, both tumor normal and tumor-only perform equally well. We find that the burden of germline classification for secondary findings is high, and the match normal itself presents its own challenges, as we showed with the adjacent normal contaminated by tumor. So in conclusion, we offer a third view in the tumor profiling debate, where both tumor-normal and tumor-only analyses are viable solutions. So when a normal sample can be obtained, it should be analyzed together with the tumor so that somatic and germline calls can be differentiated. Cancer predisposition and secondary germline findings can and some should be reported, but care must be paid to proper classification, consent, and education. When a normal sample is not available, which is often the case in clinical settings, sophisticated filters and annotations can be used to approximate somatic status, although many germline variants will still come through, as we saw today. But putative germline variants can be flagged for further testing. And finally, as you evaluate different methods, it's important to keep in mind that to do a fair comparison, you need highly optimized bioinformatic pipelines for both tumor-normal and tumor-only. And how exactly you define the set of actionable variants you analyze really affects your results. Thank you all for your attention, and I'll hand it back to Toby for the Q&A session.
Elena, thank you for that informative presentation. Before we get started on the Q&A session, I'd like to remind the audience how to submit your questions. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of your presentation window. We'll try to answer as many questions as we can. So, our first question is, having said all that, do you recommend submitting a normal with the tumor specimen?
That's a good question. So in the end, the answer is it depends. It depends what your application is. So if you are looking at a very refined, narrow set of actionable hotspot cancer mutations, like we showed, a normal might not actually be absolutely necessary for that refined set of variants. And similarly, if you're looking at mutational load or mutational burden, an application such as this, again, a normal might not be necessary to assess relative mutational load across samples. However, once you start to broaden your definition of actionable and you're interested in mutations that are not as well characterized, that's when a normal is extremely important, as we saw, to really differentiate between somatic variants and germline variants.
Thank you. Question number 2 says, you talked about SNPs, SNVs, and indels. So how does the matched normal affect somatic copy number analysis?
Great question. We are working on that currently, so we'll let you know.
Thank you. So when you described the ACE technology, you mentioned optimized coverage of the genes on the panel and on the exome. How was this accomplished?
Yeah, so we accomplished that with our ACE technology, the accuracy and content enhanced technology that I mentioned. And that consists of both additional probes and optimized probe design, as well as optimized sample library prep techniques. And we utilize a few different protocols in order to achieve uniform coverage across even those hard-to-sequence regions of the genome, like GC-rich.
Thank you. Next question says, do you require a matched normal for your clinical test?
So we do take in normals for our research products, but for our clinical test, currently, we actually don't require a normal.
Mm-hmm. Okay, so the next question coming in says, how do you differentiate possible tissue mosaics versus true tumor mutations?
Yeah, so, I mean, it depends on the matched normal you have. If you have a blood normal, then you can differentiate using that. That's a good question, though. We have thought about this in the past. If you have a blood normal, you wouldn't expect the mutation to be in the blood as well. So that's a good way to differentiate that.
Mm-hmm. Thank you. So let's see. I think we have one more question. So some other reference standards do exist, such as those available from Horizon Discovery. Why couldn't you just use those for your optimization?
Yeah. So Horizon does have a few reference standards that are great. They each contain a few important cancer variants, and we have used those. But we felt that for our large assay to really assess how we do across the entire assay, it just wasn't sufficient. So that's why we had to go about creating our own cell line mixtures and trying to curate our own gold set ourselves. In addition, the Horizon standards, they don't give you all of the variants that are present, so it was hard to assess specificity and PPV using those standards.
The next question says, can the ACE exome analysis pipeline be used for neurological panels as well, or would the parameters have to be tweaked to accommodate for that type of sample?
No, I believe, we have our ACE exome for that's used for neurology and is used for more germline things. I don't know if this question pertains to in particular cancer-related
Yeah,
neurological.
unclear from the question.
But we do have a research exome that's used for non-cancer applications as well.
Thank you. Yeah. Okay, so that was our final question. In closing, I'd like to once again thank LabRoots, as well as the sponsor, Personalis. We invite you to visit personalis.com for more information about our company and our capabilities. I'd also like to thank Dr. Helman for her informative presentation. As a reminder, you can get your continuing education credits by clicking at the bottom of your web view. And thank you, and we'll see you next time.